Hi, my name is Sandra Cangelosi. I'm a biology teacher at Torrington High School, and I work with Professor Sharon Gusky of Northwestern Community College to support and sponsor students in the high school research experience bioinformatics course, including Fhasal Ali and Logan Wilson, who will be discussing their work in this presentation. The basis of the research experience this term and in the course has been on bacteriophage annotation. In this presentation, Fhasal and Logan dive deeper into their area of interest and will be discussing their poster and research on the genomic analysis of bacteriophages Sahara and SoilAssassin. Fhasal and Logan's research focuses on the promoter regions and conserved repeats of their bacteriophages, and they utilized a number of bioinformatics programs to conduct their research in order to better identify which genes are required for survival and function. With the assistance of information afforded by PhagesDB, DNA Master, coupled with Softberry Gene Identification Program, was used to identify promoter regions of Sahara and SoilAssassin. A blast run on PhagesDB was also used to identify conserved repeats, roughly 20 to 25 base pairs long, repeated sequences indicating segments required for phage survival. The implications of Fhasal's and Logan's research are far-reaching. The host bacteria, Gordania, while not very infectious itself, is related to its cousin pathogen that causes tuberculosis. Genomic research on Gordania's bacteriophages, then, may help assist in the treatment of TB. I will now hand it over to Logan Wilson.
Hello, thank you for joining us today. My name is Logan Wilson, and I am a senior at Torrington High School. A couple of months ago, I participated in a summer research project at Northwestern Connecticut Community College. One aspect of this project was the search for bacteriophages under the CPhages Project, a national project run by the Howard Hughes Medical Institute. I was, unfortunately, unable to find any phages. Two students from the University of Pittsburgh, however, were successful. They discovered two bacteriophages, SoilAssassin, discovered by Daniel Beery, and Sahara, discovered by Sahara Grinkowitz. Both phages are in cluster CZ and subcluster CZ2 and attack the bacteria Gordonia. Gordonia bacteria, for reference, is closely related to the pathogen which causes tuberculosis. We access the genomic information of these phages through GenBank, an online database containing the genetic information of many bacteriophages. We began our analysis of the genomes of SoilAssassin and Sahara in hopes of finding promoter regions and conserved repeats, which would help us understand more about the genetics of bacteriophages.
Hi, my name is Fhasal Ali, and I thank you for your time. First, I would like to provide you with our question and our hypothesis. The question guiding our research has been, what similarities do these two bacteriophages share? And what do these similarities reveal about the importance of certain genes in Sahara and SoilAssassin? And our initial hypothesis was that by comparing our bacteriophages, promoter regions, and conserved repeats, we would identify which genes are the most important to their survival and function. In terms of our methods of evaluating this, we started by obtaining background information for both bacteriophages through PhagesDB. This provided us our information about lysine production for both phages. Afterwards, we used bprom, a program that searches for promoters. Our results were underwhelming, so we moved on to use DNA Master, the central bioinformatics software for our project. With DNA Master, we produced most of our data regarding promoters and also our data for reverse strand similarity. Lastly, we use the BLAST tool in PhagesDB, which provides data on genetic similarity, revealing our matching nucleotide regions called conserved repeats.
So now, let's run through our results. We found that 7 out of 13, or 54% of our reverse gene strands match up. Genes 30, 31, 32, 33, 35, 37, and 39 match between our phages, whereas genes 29, 38, 40, 41, 42, and 43 do not. After running our genomes through BLAST, we found that our genomes are 95% similar. This tells us that genetically, Sahara and SoilAssassin are nearly identical. In our search for promoters, we were able to find promoters for 71 out of 74 Sahara genes and 73 out of 74 SoilAssassin genes. This means that we had a 94% success rate for locating promoters in the Sahara and a 98% success rate for locating promoters in SoilAssassin. Further, we located identical promoters in Sahara's genes 9, 10, 38, and 68, which correspond with SoilAssassin's genes 9, 10, 39, and 66. In our search for conserved repeats, we found conserved repeats in 35.1% of Sahara's genes and 33.7% of SoilAssassin genes. These similar percentages indicate that the concentration of conserved sections of genomic data is relatively high. Both of our searches for conserved repeats yielded significant results within genes in 3, 15, and 28, indicating that these genes are very important to the structure or function of our respective bacteriophages. In both phages, gene 3 is a portal protein and gene 15 is a tape measure protein. Differently, gene 28's function is unidentified in Sahara, while it serves as a minor tail protein in SoilAssassin. In Sahara, we found that our conserved repeats were more concentrated at the beginning and end of our genome, whereas in SoilAssassin there was a higher concentration of conserved repeats in the middle of the genome. Lastly, we found that both bacteriophages have two genes for lysine production. In both phages, lysine A is coded for in gene 18. We found a slight difference for the production of lysine B, however, in that Sahara's gene 23 produces lysine B, whereas SoilAssassin's gene 24 produces lysine B.
To summarize, genes 3, 15, and 28 had a high concentration of conserved repeats, leading us to conclude that they are important to our phages. Gene 9 had matching promoters and also codes for the head-to-tail stopper. After doing a bit of research, we found that this gene impacts the proper insertion of DNA from viruses and bacteria. Lastly, gene 18 may perhaps be the most notable gene, as not only did it include an incredible amount of conserved repeats, it directly codes for lysine A in both bacteriophages. Therefore, we can conclude that genes 3, 9, 15, 18, and 28 are integral to the function of our phages.
Our research highlights the importance of lysine production within bacteriophages. Lysine directly attack the cell walls of bacteria, so understanding lysine-producing bacteriophages will provide a viable alternative to antibiotics. Bacteriophages only target one type of cell, the bacteria that they are designed to attack. This means that phages can be used as a specialized treatment against certain pathogens. One limitation to our research, however, is that it focuses on pathogens which attack Gordonia. Gordonia is not a common pathogen, so our research does not directly fight against any diseases. However, because Gordonia is closely related to the pathogen causing tuberculosis, our research may be applicable in the fight against TB. Lastly, our research leaves some unanswered questions. Why do Sahara's genes 10, 38, and 68 and SoilAssassin's genes 10 and 66 have identical promoters but no identified function? Also, why does SoilAssassin have a defined function for gene 28 but Sahara does not? These questions leave opportunities for further research. Luckily, our identification of promoter positions leaves future researchers with a perfect head start.